Hi, everybody. Now we're going to talk about medical microbiology and how, though we have so many beneficial microbes, those that are bad are really, really bad. And so we can look at the bad microbes. The microbes that cause disease are typically called pathogens. And one specific pathogen, influenza, in 1918 to 1919 killed more Americans than both world wars plus a plethora of other wars all combined. Um, so it's very dangerous. These diseases can be very dangerous. And so this is a table or a figure that shows in 1900 the leading causes of death. And down here shows the leading causes of death close to 2000. So up here in the early 1900s, the leading cause of death was pneumonia. Down here it's heart disease. The second most common cause of death was tuberculosis, another respiratory disease. Second most common cause of death is cancer. Then we had diarrhea and enteritis. Diarrhea isn't even on this list. So the first three were all caused by microbes in the 1900s. By the end of the 1900s, I'm looking down here, pneumonia and influenza don't even hit the top 5. So they're very uncommon. They don't cause many diseases, or they don't cause many deaths. I won't say diseases, deaths, as they did in the past. And so this is really important. What this means is, or what this says is that our medical system has become very advanced. We now have doctors and nurses that know how to follow aseptic techniques in hospitals to keep the patients from getting sick. We know how to treat diseases. We know what diseases are caused from, so what is causing this disease, and so we can target that disease. So these are all really important. We also have antibiotics, which we didn't have in 1900, and we have vaccination. So these are huge. So let's look before the 1900s. Let's look what people, or let's look at some of the individuals that established medical microbiology. Augustine Obasi, back in the 1800s, he demonstrated that silkworm disease was due to a fungal infection. And so he was able to do this, even though at this point people still weren't sure what or if microbes cause disease. They didn't know at this point. And so here he studied these silkworms, and so I just found these pictures off the internet. This is Agostino. This is a healthy silkworm. So this is what a silkworm with silkworm diseases looks like. It has this fungus that surrounds it and basically keeps the worm from being able to breathe properly, and the worm dies. It eats the worm and the worm dies. And what he was doing is he had, if a worm had silkworm disease, he would separate it from the other worms. But even if the worm didn't look like it had anything on it, if it was next to this worm, then the worm would get sick. And he realized that there were microscopic structures likely coming from this fungal coat and getting on the worm. And so that's where he demonstrated that the microbe, the fungus, was actually causing this disease. Joseph Lister then developed a system of antiseptic surgery. What he did was he produced a carboxylic acid cleaner. So this little machine sprays carboxylic acid out, and that sanitized, sterilized the environment. It didn't really, I won't say it. It was used to clean the environment, okay, so not sanitize, and it didn't really sterilize because there's still living things here. but it cleaned the environment. So he made his surgeons wash their hands before every surgery, and he would spray this carboxylic acid on the table, on all the tools that were going to be used in the surgery, and then he sprayed it on the patient as the patient was being operated on. This increased survival of surgery by 50%, or by more than 50%. He also implemented changing of sheets in the hospital. So prior to this, when people died in the hospital, or if people were ill and they got better and then they went home, unless the sheets were very, very filthy, they were not changed. They looked at the sheets. If the sheets looked good, then they just left them. But we know that microbes are super small, and so you wouldn't see them on the sheets, but they could still cause infection in otherwise immunocompromised individuals that were now coming into the hospital and sleeping in that same bed. So he did a lot for the field of medical microbiology. Robert Cook then, who is right here, there we go. He was the one who established a relationship between specific pathogens and specific diseases. So he actually used criteria that his teacher, so his professor, had developed, and drew on them to make what we now call Cook's postulates, which are still used today. And what these do is they link specific microbes to specific diseases. So here are the postulates. There are 4 postulates, and if all 4 postulates are met, then we know the causative agent of a disease. So in any disease, the microorganism that causes the disease must be present, but it must be absent in healthy individuals. That microbe must be able to be isolated and grown on a pure culture. That same disease must result if the isolated microbe is inoculated into a healthy host. And the same microbe must again be isolated from the newly diseased host. So here is basically what's going on. You have a microbe that causes a disease, you think, in this case, let's say tuberculosis. I think Mycobacterium. Tuberculosis causes tuberculosis. So if I take Mycobacterium from an individual that is diseased with tuberculosis, if I take their sputum and I isolate the tuberculosis and I infect another organism, and that organism gets tuberculosis, and I can isolate the tuberculosis again, that allows me to know that Mycobacterium tuberculosis causes tuberculosis. But there are limitations. So an example, certain organisms can't be grown in a pure culture. Mycobacterium leprae only grows inside of animals. And so if it's growing inside of an animal, you can't get it in a pure culture. We can't use humans in completing postulates. That would be unethical. And now that we have molecular and genetic techniques, this may eventually get replaced. But Cook's postulates did a lot for us. So this, we had to be able to develop mechanisms for isolating bacteria. So a lot of our aseptic technique. And we had to be able to grow bacteria on a hard substance, on something that was solid. And so we initially used broths, but now we have added agar to those broths to produce a solid agarose gel medium. Actually, agar has been used for a long time in, well, in the food industry. So here we're using it to make solid nutrient agars and stuff for bacteria to grow. But Franny Hesse actually used this in making her jam so that they wouldn't liquefy in the hot sun. And she's the one who suggested they try it in their nutrient broth, and it worked. With the development of agar, we had to use plates or dishes, so we developed petri dishes or petri plates. And now we have nutrient broth and agar that we use. And then we have mechanisms for isolating those organisms. Right, so now we know that diseases are caused by, or that some diseases are caused by microbes, and we can even identify the disease to the microbe. So this brings us to immunological studies, which I'm going to get to in our next video. Bye.